Hi team, today we're going to talk about linkage. And to start to think about that, I'd like you to consider this pedigree or family tree. In this family tree, each male is shown as a square and each female is shown as a circle. And then individuals who are carrying our trait of interest, that is an autosomal dominant trait for it doesn't matter what, we also know, so we know their phenotype, but then we also know their genotype at this gene A. So take a few minutes and think about which allele of A is most closely associated with the trait we're interested in. Pause the video, look at their genotypes, and let's come up with an answer. Hopefully you observed that almost every individual who has this trait also carries the A1 allele. Now, not everyone has the A1 allele who is showing our trait, but most of them do. So if we were to draw this on a chromosome, you might imagine that the trait we're interested in here is located at one point on a chromosome, and the A1 allele is located very nearby it. So that when the A1 allele is inherited, so is this trait or this mutation. So we're going to talk a little bit more about these ideas of things being linked on a chromosome today. In this first set of slides, we're going to be talking about linkage. We're going to be talking about the idea of coupling or repulsion. Another way of saying this is cis and trans, and this is in reference to the different alleles of a gene. We're also going to be talking about making a map of our chromosome. And last but not least, we're going to be talking about linkage groups that affect a particular trait. Today we're going to focus not just on linkage, but thinking about making a map of the chromosomes. So here are 3 different maps of London. I think you can see the river running through all of them. And you can see that they have varying degrees of complexity. So we're going to try and make a simplified version, like this one on the right, of a very complicated reality, and that is the structure of chromosomes. For example, here I have 2 chromosome maps, and if we focus on this one on the right, this is showing us a Drosophila chromosome. That is a chromosome from fruit fly. Along the length of this chromosome, you can see that there are any number of genes dealing with body color, the bristles on the fly, shape and color of eyes. So this chromosome has many, many genes located on it. And this map represents a simplified view of a complex chromosome. Now on the left-hand side, we're considering the human X chromosome. And this is a simplified map, though it is much more complex than the Drosophila chromosome. You can see we have the location indicated of multiple different disorders or diseases, and they're localized to small regions on the X chromosome. So even in, let's say, this tiny region at the bottom of the X chromosome, all of these disorders have been associated with genes or mutations in this tiny region of the chromosome. There's a lot going on here, and so it's really important for us to be able to simplify and sort of make a map of what's going on at the genetic level. In order to think about linkage maps or chromosome maps, we need to consider recombination. Let's consider an individual who has this dihybrid genotype, big A, big B, little b. Now, if the A and B alleles are independently assorting, you can imagine that the gametes we'd produce could be big A, big B, big A, little b, big B, little b. And all 4 of these gametes would occur with equal proportions. That is, if we observe a big A in the gametes, 50% of the time the big A will be with big B, and 50% of the time it will be with little b. either one of these combinations is equally likely because big A and big B are moving independently of one another. The complete opposite of that would be every time we see big A, we also see big B. Every time we have little a in the gametes, we also have little b. And we never see big A, little b. We never see little a, big B. Big A and big B are completely linked on the chromosome. That might look something like this. If we had a chromosome where big A and big B were so close together, we never saw them separate during recombination. A more likely scenario would be that big A and big B are neighbors on the chromosome, so that when recombination occurs, I'll draw a second chromosome here that has the other two alleles, little a and little b. Some proportion of the time, we can have crossing over. So we're going to end up with more big A, big B gametes, more little a, little b gametes, and then few where this recombination event will take place. And we end up having big A with little b gametes, and also few of the little a, big B gametes produced by recombination. Okay. Let's think about genetic linkage and recombination in another way. The simplest way to think about this is that during meiosis, we have 4 chromosomes lining up during meiosis 1 and then separating ultimately into the gametes during meiosis 2. If there's no crossing over, each one of these sister chromatids will end up into a separate gamete, shown over here on the right. If there is crossing over between these two homologous chromosomes, and that's shown right here, we're going to end up with some chromosomes that are non-recombinant on the outside here. That is, chromosomes that haven't had crossing over, so no crossing over. And then some chromosomes, these two in the middle, that are recombinant, that have experienced crossing over. You can see now we have a new combination of alleles. So recombinant chromosomes have new combinations of alleles. For example, in the original individual over here on the left, here's our original person, they had one chromosome with big A and big B, and a separate chromosome with little a and little b. But in the gametes, we now have a chromosome with big A and little b, a chromosome with little a and big B, as well as the original chromosomes big A, big B, and little a, little b. So we've got a new combination of alleles, and that's caused by recombination. Which one of these will be more common? Well, the most common is to have no crossing over. So it's going to be more common for us to have these non-recombinant gametes. More common to have non-recombinant gametes. It's going to be less common to have recombination occur. The last set of terms I'd like us to think about is the idea of coupling and repulsion, also called cis and trans. Now, in cis, this refers to an arrangement of the alleles, like the red and blue chromosomes shown above, where one chromosome has big A and big B, the other chromosome has little a and little b on it. This combination of alleles would make up the genotype big A, little a, big B, little b. They're heterozygous for both genes. But you can imagine you could still have this big A, big B, little b genotype if your chromosomes look like this. Big A with little b and little a with big B. The difference between these two chromosomal arrangements is only how they're arranged. So on the right-hand side here, I would say that these chromosomes are in trans. That is, they're in repulsion. Whereas on the left, we have cis or coupling. And these terms mean the same thing, and I'll use them interchangeably. So in organic chemistry, also if you think about double bonds and fatty acids, cis means same. Trans means across or opposite. Here we have, in cis, we have the same alleles, that is a big A and a big B on one chromosome. In the trans chromosomes, we have a big A and a little b. The alleles are different from one another. Or another way to think about it is the big A and the big B are opposite or across from one another. So coupling and repulsion is just a way of naming the orientation of the chromosomes. So that's what coupling and repulsion are. They are naming the orientation of the chromosomes, or rather of the alleles on the chromosomes. So we've talked about genes and their linkage on the chromosomes. Using that information, I'd like us to draw now a chromosomal map of a couple of genes. In order to do that, we need to set aside some terminology first. First of all, a map unit refers to 1% recombination. And by definition, 1% recombination is 1 centimorgan. So map unit equals centimorgan equals percentage recombination. So let's draw a map starting with genes A and B. We can see that A and B are 5 centimorgans apart. So let's draw A here and B here. That means we can draw 5 centimorgans between A and B on the map. Because centimorgans are equivalent to recombination percentage, we can expect to see recombination occurring 5% of the time between A and B. And we'll do some more practice problems to help you think about what that actually means. Now, between B and C, we have 10 centimorgans, and A to C, we have 15 centimorgans. We're going to have to make our chromosome longer, folks. Let's put C out here on the right-hand side. We can have now 10 centimorgans between B and C, and then a total of 15 centimorgans between A and C. Pretty straightforward. Let's make it more complex. I'm going to redraw our original chromosome with A, B, and C being 5 and 10 centimorgans apart. So all of this data is already included on our map. Now we have to add in a fourth gene, gene D. Consider this data and where we should put gene D on the map. Pause the video and let's come up with an answer. Well, let's think about this. B to D is 13 centimorgans. Okay, so maybe it's way over here. And then C to D is 23 centimorgans. Oh, it's further from C than it is from B. Further from C than from B. I would put D over here on this side. So if this is 13 centimorgans, then this would be 13 plus 10, 23 centimorgans. Nice. What's the expected rate of recombination between A and D? Well, we can use math to solve that. If this is 5, then this must be 13 minus 5. It's going to be 8 centimorgans, and we know that 1 centimorgan is equal to 1 percentage of recombination. So there's going to be 8% recombination between A and D. All right, tune into my next video where we think a little bit more about what this 8% recombination would look like and think more about what offspring after recombination might have as their genotype. See you soon.